Hi, this is GCSE 9 to 1 Crammers. It's never too late to start revising, and this way you can do it anywhere. Listen to these questions as many times as you need until the answers pop into your head. Today we're revising Biology Topic 1A, Culturing Microorganisms, covering what are microorganisms, how microorganisms are prevented from entering the body, equipment for growing microorganisms, how to work aseptically, investigating the effects of antibiotics, and revision questions. What are microorganisms? Microbes, like bacteria and viruses. What do microbes need to survive? Water, oxygen, and warmth. Describe 5 ways the body prevents the entry of microorganisms. Skin forms a barrier, which produces antimicrobial secretions to kill off and stop microorganisms. Nose hair traps particles, which may have pathogens, which we get rid of when we sneeze. Bronchi have cilia, which wafts mucus to the throat to be swallowed. The bronchi and trachea both have mucus, which traps pathogens which we swallow or cough out. And hydrochloric acid, which is stomach acid, kills pathogens in food that we eat when it enters the stomach. What equipment is needed to grow microorganisms like bacteria in a lab? An agar plate, which is a petri dish, with a culture medium, e.g. agar jelly, set in it. An inoculating loop, used to transfer microorganisms onto the agar jelly, and a Bunsen burner, used to sterilize the inoculating loop. You will also need paper discs, soaked in different types and different concentrations of antibiotics, antiseptics, and disinfectants. What does it mean to work aseptically? Everything must be sterilized before use to kill unwanted microorganisms and stop contamination. Explain how to work aseptically. Sterilize the petri dish by heating it up to a high temperature. Sterilize the agar jelly. Heat it up and then pour it into the petri dish and let it cool and set. Sterilize the inoculating loop by passing it through a hot flame. This must be done before and after transferring bacteria onto the agar jelly. Storing the culture. Lightly tape the lid on. This is because it stops microorganisms from the air getting in. Then turn the petri dish upside down, because this stops drops of condensation falling onto the agar surface. Investigating the effects of antibiotics. You need a bacteria sample applied to the agar jelly, a paper disc soaked in antibiotic, and a control disc soaked in sterile water, not antibiotic. Then you store the sample plate to let the bacteria grow. Should an inhibition zone appear, which is the white area that forms around the paper disc, this shows the antibiotic is effective and has stopped any bacteria growing in this area. You will see the control disc has no inhibition zone and bacteria is growing around it. This proves the antibiotic stopped the bacteria growth. What do microbes need to survive? Water and oxygen and warmth. What temperature would be most suitable for growing bacteria in classroom conditions? 25 degrees centigrade. Why is it important that plates aren't stored above 25 degrees centigrade? To reduce the chance of harmful pathogens growing. What temperature would be most suitable for growing bacteria in industrial conditions? 40 degrees centigrade. What is the advantage of using the temperature of 40 degrees centigrade? Microorganisms grow reproduce and act faster, so you see the results faster. What is a control disc? A paper disc that has not been soaked in antibiotic, but instead has been soaked in sterile water. What is the purpose of a control disc? It shows that the lack of growth around the antibiotic discs is due to the effect of the antibiotics and not something else. What result would you expect to see if an antibiotic was not effective against a strain of bacteria? There would be no inhibition zone around the paper disc with the antibiotic on. How would you get an even covering of bacteria on the medium in the agar plate? Spread the bacteria evenly using an inoculating loop. What could you conclude from the results of the investigation if there is no inhibition zone around the antibiotic you are testing? That type of antibiotic was not effective in preventing the bacteria growth. What result would you expect to see if an antibiotic was effective against a strain of bacteria? A large inhibition zone around the antibiotic, stopping the bacteria from growing. What measurement would the scientist need to take to calculate the area where no bacteria have grown? The radius of the inhibition zone where no bacteria have grown. Give two variables that should have been controlled in order to make the results of this investigation valid. The size of the paper discs and the concentration of the antibiotic. How would you prevent contamination of bacterial cultures? Use a sterilized inoculating loop by passing it through a flame. Use a sterilized petri dish. Tape the lids onto the petri dishes. Store the petri dishes upside down and use sterilized agar jelly. Why is it important that cultures are not contaminated? Because contamination by unwanted microorganisms could cause the growth of pathogens and may have affected the results of the experiment. The diagram below shows the inhibition zones produced by antibiotics A. penicillin and B. streptomycin. Find the areas of the inhibition zones and state your conclusion. The formula to find the area where the bacteria have grown is area equals pi r squared. You also need to find the radius, divide the diameter of each zone by 2. First, we will find the radius of A, which equals 14 divided by 2. Do the maths, and it equals 7 millimeters. Now we know the radius, we can work out the area of A, which equals pi times 7 squared. Do the maths, and that equals 154 millimeters squared. Now we work out the radius of B, which equals 20 divided by 2. Do the maths, and that equals 10 millimeters. Now we can work out the area of A, which equals pi times 10 squared. Do the maths, and that equals 314 millimeters squared. Looking at the areas for Zone A compared with Zone B, we can tell that 154 times 2 equals 308. This means that Zone B is roughly twice the size of Zone A. So the conclusion would be antibiotic B, streptomycin, is more efficient than antibiotic A, penicillin, when fighting this type of bacteria because the size of inhibition zone B is roughly twice the size of zone A. In this video you've learnt about the harmful microorganisms and how to prevent them from entering the body, how to work aseptically, and how to go about investigating the effectiveness of antibiotics on different bacteria. Take time to learn this topic so you can nail those questions in the Biology GCSE Paper 1 exam. The key to exam success is learning, consolidation, and keeping this topic fresh in your mind. How do you do that? Repetition, repetition and repetition. If you go over these enough times, you'll be able to answer any questions on this topic confidently in your exam. This is GCSE 9-1 Crammers, and if you like the content of this video, give us a thumbs up. And if you have a question or need help, give us a comment below. And don't forget, smash that subscribe button if you want more Crammers videos for your exam success. Thanks for watching and see you again soon.